Thank you so much. Well, welcome back. My name is Arnold Levine, and it's a real pleasure to be able to welcome you to the Institute for Advanced Studies, which is hosting this meeting for the Cancer Institute of New Jersey. The next 2 hours, we're going to hear 3 talks on the general area of the new genomics, identification of risk. And these 3 talks will actually address 3 related ways or methods being developed in research laboratories that are just being applied in the clinic that, in fact, risk could be assessed. Laura Van Veer, the 3rd speaker, will talk about the use of transcriptional profiling of messenger RNA species in cancer cells to make some predictions about the outcome of those cancers and indeed the treatment of those cancers, which is, of course, one of the long-term goals. Tim Rebick will continue the discussion that we actually began in the first hour on BRCA1 and talk about the variables that modify BRCA1 and the use of human genetics to understand the disease and to understand risk in greater and greater detail. And I'll talk about the use of single nucleotide polymorphisms, which we heard about in the first hour, on a different signal transduction pathway, the p53 pathway, which is a pathway that responds to stress. And the p53 pathway is one of our major deterrents for the development of cancer. A good example of how the p53 pathway works is that as we go outside for lunch and we walk over to the cafeteria, each of us will be exposed to a certain amount of sunlight, not terribly threatening, but in some cases that sunlight will react with the DNA of our skin cells, and that reaction will provide energy which will change a base, and a mutation will ensue in our skin cell. This mutation will be recognized by one of our gene products, the p53 gene product. And in recognizing the mutation as damage DNA, it will kill the skin cell. p53 will activate in response to the DNA damage and kill the cell. And activation in this case means p53 protein concentration increases, and it acts as a transcription factor to turn on a program of genes resulting in death or apoptosis. And in that way, we constantly, in our lungs, in our intestine, in our skin, we constantly clear damaged cells, which could have the high probability to go on to carrying mutations, somatic mutations, not genetically inherited mutations, that have the high probability of going on and developing cancer over a lifetime. And so this surveillance system against stress is present in us over a lifetime. And the first slide here just shows the system that's in place in our cells at the molecular level for this stress response. And at the top, a list of different kinds of stresses that activate p53 are found. This is the one I focused on, DNA damage with UV light, but reactive oxygen species or gamma radiation from the cosmos all damage DNA constantly over our lifetime. Hypoxia, the lack of oxygen in certain tissues, will be a stress signal, and the body responds to hypoxia, and one of the ways is it activates cells. It activates the p53 protein shown here, and in so activating through this pathway the p53 protein, it'll kill cells undergoing hypoxia that make mistakes in the replication of the cells. Nutrition, ribonucleoside triphosphate depletion, so glucose starvation is recognized as a stress by this system. Spindle damage, that's the structure of the spindle which segregates chromosomes equally into 2 cells. If that's damaged, you get unequal segregation. Nitric oxide is really quite interesting. Nitric oxide is a signal of inflammatory response. So even infections that the immune response is responding to communicate in some way with this signal transduction pathway that can give rise to cell death. Nutrient deprivation, I mentioned, heat or cold shock, or even the mutation of certain oncogenes that predispose us to cancer in somatic cells are recognized as an inappropriate signal, and p53 responds. So this is our first response to all sorts of stresses, and these stresses result in mistakes being made during cell division, and those mistakes or mutations being made in cell division raise by about 1,000-fold our predisposition to get cancer because of that. And what protects us against that is that each of these stresses is signaled in some way to p53, which, as I say, activates. That means increases its concentration, becomes a transcription factor. And the outcome of that is one of three things: apoptosis, which is cell death; senescence, which is a permanent cessation of cell division; or cell cycle arrest, which is a transient cessation of cell division. And cell cycle arrest allows for repair of these stresses and then re-entry into the cell cycle in division. Now, the p53 protein is actually regulated very tightly in the cell, and its major regulator is a gene I'll talk a lot about. It's called MDM2. MDM2 is a ubiquitin ligase for p53, so it polyubiquitinates it. That sends it to the proteasome and trashes it. So MDM2 is a negative regulator of p53. It lowers its levels. But this is actually in an autoregulatory loop that keeps the levels constant. p53, when it's activated, actually transcribes the MDM2 gene. So there's a positive back to MDM2 and a negative regulation of p53. This sets up a rather natural oscillator in the cell between these two proteins, and they oscillate about 180 degrees out of phase in the cell, and that oscillation can be changed by stress, and the change leading to the outcomes that are found here. Now, we've been searching, and we as Gareth Bond and Wenwei Hu have been searching for a number of years now for single nucleotide polymorphisms that change the activity of these proteins. And these are found as inherited forms of polymorphisms, a single base change, that may change the activity very little, like 40% or 60%, but this is such a central feature of our stress response that over a lifetime, a 40% difference in activity may add up. And what we found here on the p53 gene is a polymorphism in the coding region of the gene at codon 72, amino acid 72. Some of us, most of us in the audience, have arginine at that spot, and some of us have proline at that spot. And those of us that have arginine have about a 40 to 60% increased efficiency of apoptosis. And so the ARG homozygotes in the audience are a little bit more efficient at responding to sunlight or gamma radiation or nutrient deprivation or spindle damage, a little more efficient than those that have proline at both chromosomes in that spot. There's, of course, 2 copies, and there could be heterozygotes of pro-ARG. Similarly, there's a second single nucleotide polymorphism I'll talk a lot about, although I'll mention this, in MDM2, which is not in the coding region, but is in the first intron of the MDM2 gene. And those of us that have GG at that position, the homozygotes, make about 2-fold or 3-fold more MDM2 messenger RNA. So this is in the promoter of the gene. And they make more messenger RNA, and they make more MDM2 protein. Those of us that have TT at this particular spot, in fact, make less protein. So some people in the audience, if we were to take blood tests, have about 2-fold higher level MDM2 than others. The GGs are high, the TTs are low. And that, of course, has an impact on the level of p53. And together, these polymorphisms have an even bigger impact. And so what I want to do is show you the impact of these polymorphisms on the occurrence of cancers and what we found out about them. And in particular, my take-home message here is that the estrogen receptor actually also binds to the MDM2 gene. So this polymorphism is going to be particularly important in premenopausal females that have estrogen receptors, of course, and have estrogen. This is an example of the first study we carried out with sarcomas from Germany and Li-Fraumeni patients. Li-Fraumeni patients are patients described first by 2 epidemiologists, Li and Fraumeni, that have very high incidence of cancer in their families. It's actually described as a proband that shows up with a sarcoma, and that there's at least 2 first-degree relatives that have cancer with the proband. So this is a very high incidence mutation. And people that have germline p53 mutations then develop cancer with this virtually 100% penetrance, 98% penetrance over a lifetime, and they develop cancer at an early age. It's quite like BRCA1 in that sense. So here's the example of someone who's a homozygote of TT for the MDM2 gene. You make very little MDM2, you make a lot of p53. p53 is protecting us against stress. Here's the GG. We know that there's a transcription factor, SP1, that binds to it. This extra transcription factor makes large amounts of MDM2 and a very little p53. And then if we look at the patients that have cancer, this is the fraction. There is in this group about 165 patients with sarcomas from Germany, all admitted to the same hospital, all treated the same way, all diagnosed the same way. We look at the age of onset as an indication of the impact of high MDM2 GG or low MDM2 TT versus the frequency of getting the tumor. That is to say, 100% is, of course, all 160 of these patients. You can see in blue line here are the TTs, and in green line here are the GGs. You can see that there's about an 8 to 10 year earlier age of onset. And we interpret that in the following way. If you have a system that protects you against stress and protects you against cancer, and over a lifetime you have high p53, you'll develop cancer in this world where everyone had cancer, right? Develop cancer at a later age than if you have low levels of protection over your lifetime from sunlight and gamma radiation and reactive oxygen species. Now we turned our attention, once this is actually published in 2004, this work, and we turned our attention to a different kind of tumor that was very enlightening. This was a diffuse large B-cell lymphoma. This is a study of about 360 patients from Sloan Kettering, carried out with Ken Offit and his colleagues. And here are, this is just the patient population, stratified into females and males to make a point here. Age of onset versus the fraction of the population up to 100% that have developed cancer at that age. And what you can notice is that this is a sexually dimorphic cancer. Males get it a little earlier than females do, and this was well known from the literature, and this is our patient population. Now, it was this group that we then genotyped for GG or TT in the MDM2 gene. So we're going to now take this patient population and stratify it, and we're going to stratify it and separate males and females because we know it's a sexually dimorphic disease. Here's an example of that. This is the female population. If they are GG, you can see that you get an earlier age of onset, TT in red, GG in blue. Earlier age of onset, in fact, the average is 68 for the TT's age, 55 for the GG's. And you can see the curve, I'll come back to this, is biphasic and the same after a certain age. Probability that this is by chance is only about 2%. These are the males that are TT and GG, and the bit of a surprise at first when we saw it is that there's no difference between this, 58 and 60 in the males. So this is a single nucleotide polymorphism that acts in females but not males. And note the shape of the curve. It has an earlier age of onset prior to about 55 years old, 51 years old. That's the age at menopause where the estrogen levels start to decline, and when that happens to postmenopausal women, the onset is about the same, whether you're TT or GG. And this led to the hypothesis that this was a single nucleotide polymorphism that was modified by estrogen in some way. That that's in fact a very good hypothesis is shown by looking at the DNA sequence here of the polymorphism. Here's the T to G polymorphism. This is the DNA sequence in the first intron of the MDM2 gene. Here's the T to G polymorphism, and right 10 base pairs away is an estrogen receptor DNA binding site. And that estrogen receptor DNA binding site has been subsequently shown to bind the estrogen receptor by chromatin immunoprecipitation. And quite interestingly, the GG form attracts a transcription factor SP1, which is well known to interact with the estrogen receptor, and that is responsible for raising the level of the MDM2 gene. So This is an example of a complex genetic trait. While we all inherit it, only premenopausal females have a phenotype because the estrogen receptor works closely with the single nucleotide polymorphism. This is a consequence of that explanation. This is a study first carried out by Louise Strong and Gigi Lozano at MD Anderson, and they published this a year before we found the polymorphism in 2003, and it examines the sexual dimorphism found in Li-Fraumeni patients, right? So these are 88 patients that have inherited a p53 mutation. So this is an interesting group because you only have one copy of p53, so you make half the concentration of p53. And then if you have GG, you're going to have high MDM2 with half the concentration of p53, TT low MDM2. So here's the Louise Strong data that says that the age of onset of the first cancer in these patients here at 20, 30, 40, or 50, these decades, for females and males, is led in every age by females. So by the age of 50, 93% of the females have had first tumors because of the mutation in the p53 gene, but only 68% of the males. So this shows that females are affected by age of onset much more rapidly than males, and the number of, actually what's not shown here is the number of independent tumors is much higher in females than males. And here we look at the males. This is a total group of 30, right? And their average age of onset of 20 years, right? And the females, a total group of 18, and their average age of onset is 18 years. So if you look at the total, there's not much in the way of difference here. And then we look at the TTs, it's 20 for males, and the GGs and TGs, it's 19, no difference in males. But 24 and 10 years, there's a 14-year difference with about a 0.02% probability, 0.2% probability here that this is a real difference. And so much of the sexual dimorphism that's seen in Li-Fraumeni families can be explained by this particular single nucleotide polymorphism. And let me say, there have been now 4 independent studies that have confirmed the role of this single nucleotide polymorphism in the Li-Fraumeni families. I'm going to move fast and get to lung cancer. This is a study carried out by Zhang and his colleagues in China, and then a second study looking at lung cancer by Lind and her colleagues in Norway. And Zhang had about 1,200 patients with lung cancer, and then he had about 1,200 controls that did not have lung cancer. And the odds ratio then is to just place how many of the individuals that had lung cancer were GG in the MDM2 over how many individuals in the control group. And so this gives an odds ratio of contracting lung cancer and the impact of GG upon that ratio. So you can see in the Zhang study, the MDM2 GG form, that's high MDM2, low p53, gave an odds ratio of about 1.8 or so. Now we switch to the polymorphism in the p53 gene, which we know is just downstream from MDM2, right? And we have an odds ratio of about 1.4. And now the power of combining genetics in exactly the same pathway, because he had enough numbers, you could look at GGs and pros together. So this is poor apoptosis in p53, high MDM2, and the odds ratio now goes up to 4.5. And then environmental impact. Remember, smoking causes mutations in the lungs. It's a mutagen, benzpyrene in smoke, cigarette smoke, reacts with DNA, causes mutations in the lungs. So those who have poor genetic risk, but also are smokers, add to this odds ratio of about 10, right? This, I think, although it takes a very large studies to see, makes a few really important points that should be emphasized. One is we're now on the verge of looking at the interaction of genes with environment, and we're looking at it in a rational way because we understand about the stress response pathway. We understand the relationship between two genes in the stress response pathway, their polymorphisms, and we understand the environmental hazard of smoking, and we can quantify that. Right? Now, this work was confirmed in another way by Lind and her colleagues in Norway. They looked at the odds ratio for all men and women lung cancer patients who had GG, and they found a ratio of 1.62, and that's about the same as 1.83, and confirms that as an independent study. But then they stratified females only in the population, and the odds ratio went up to 4. Again, showing that here's a really good example of early onset lung cancer in premenopausal women having a big impact from this single nucleotide polymorphism. Okay. Now, one of the things that one does when you have single nucleotide polymorphisms like this is you want to examine the single nucleotide polymorphisms from the whole gene. So I'm going to introduce you to the MDM2 gene and tell you about all of the polymorphisms in the gene. This is a gene that's about 32,000 base pairs long, so it's a small percentage of the human genome. It's, like many genes, divided into exons, which are in purple, and introns, which are in green. Remember, I said the first intron had the SNP 309, or T to G change that we've been focusing on, and there it is. But there are many changes that are different in the population. Right? The population at large could be different at each of these positions, A or G, C or T. And there's 11 across here that were carried out in a SNP analysis of about 100 individuals by Solera and their colleagues in both African American and Caucasian populations. And then by collaborators of ours at the Sheba Cancer Institute in Israel, 6 of these SNPs were looked at in Ashkenazi Jewish populations. And then what one can do is to look at the stratification of the types of single nucleotide polymorphisms in a population. So we're now going to literally reproduce populations in a virtual sense, reproduce populations and what the order of these polymorphisms are across the gene, and you'll see why that's informative in a second. So this complex slide can be easily explained. These are African Americans. Here's the T's, and here's the G's that I've told you about for SNP 309. Right away, you can tell that African Americans, about 90% of them are T, and about 10% are G. And then we just add polymorphisms on either side, and we continue doing that till we get down here to all 11 single nucleotide polymorphisms. These are all T's. But now you can see they come in lots of combinations. 6% of the African American population is this order of polymorphisms, 5% this, 11% that, 12%, 20%, 12, 20. And then there's just a single haplotype or string of polymorphisms in the G allele. Now this tells us a lot. Think about what happened. The humans arise about 100 or 150,000 years ago in Africa. And the population's very homogeneous. Everyone has the same sequence. And then with time, you diversify. And that's where mutations start to occur, making single nucleotide polymorphisms, changes in the DNA, and populations breed and they get fixed in. And then recombination tests all of the possible combinations. So when you see a structure like this that has 1, 2, 3, 4, 5, 6, 7, 8 different haplotypes, you know that's the old or original haplotype. It took a long time to make the changes and recombine. You can tell the relationships between these. You can often tell the ancestor haplotype, right? And in fact, in Africa, Africans have only this diverse set of haplotypes. They don't have this. And this is a single haplotype that tells you the G is a recent mutation that occurred. And in fact, when you now look at Caucasians in the United States, you see the same structure, 50, except for the percentages, 57% and 43%. Look at this, there's all the haplotypes of T and a single G haplotype. And this single G haplotype in Caucasians is identical to the haplotype in African Americans. So this is likely due to an admixture of Caucasian chromosomes in African Americans. And when we go to Africa and look at this, we don't see this at a high frequency. Right? So this suggests that the T allele is the old allele, It's in 90% or up to 100% of Africans. It's due to an admixture in Caucasians. And Caucasians have moved up to 43% of the G allele. Now, if the G allele is so new, why is it at such a high percentage? If it was a new allele, you'd expect it to be rare. And it is rare in African American populations, but it's 43% in Caucasians in the United States. And in Ashkenazi Jewish populations, it's 53%. Right? Now, there are a couple of possible explanations. One is a bottleneck, right? If there was suddenly a very few group of people with high G content that started mating and they made a whole population, that could explain why G is so high. And the Ashkenazi Jewish population undoubtedly went through a bottleneck. But that's not true of the Caucasian populations or the Asian populations, which have 43 to 46% G content. So the other possibility is positive selection of a gene. What's the possibility that this is so high because it's favorable in the human population? And if it's favorable, why would it be the pro-cancer phenotype? Right. Now, we wanted to consider this, and Wenwei Hu, a colleague in the laboratory, began an investigation of the possible role of this gene in one of the two reasons to positively select it. Either it makes us resistant to infectious diseases, which would allow us to breed more often, or it plays a role in fecundity, the number of offspring we have. And as you'll see, the surprise is that p53 plays a central role in fecundity in mice and possibly and probably humans. So let me introduce you to that by telling you that p53, which is a transcription factor, regulates a gene called LIF, which is essential to induce a receptive uterus for implantation. So implantation and decidualization don't occur in the absence of LIF in mice, and that's only a female-specific infertility. Males that don't have this gene have no impact. Implantation is rescued by an injection of LIF in LIF minus mice, and the expression of LIF is coincident with the onset of implantation, as you'll see in a minute. So here's a gene regulated by p53 that is essential for implantation and fecundity. Right? And we can take a look at that. This demonstrates that p53 really regulates this gene. There's the sequences in the gene that actually bind the p53 protein, and you can see that by a chromatin immunoprecipitation here. You can see it in human cells. This is mouse, this is human. See it in mouse cells by a chromatin immunoprecipitation. And here we have taken these sequences, put it in front of a reporter gene, luciferase, and p53 induces in each of these cases, p53 induces the transcription of the gene. So this is a gene regulated by p53, presumably, in fact, in response to stress, and in fact, as you'll see, in response to copulation in mice. This is the study really carried out by Angie Tereski, the technician who runs the animal facility in Wenwei Hu, of the fecundity of mice in the presence or absence of the p53 gene. So this is in a black 6 background. This is an inbred mouse background. Males and females, on the average, have a litter size of about 6.7. And the fertility rate, that is to say, how productive the number of matings are, if you put one male and 3 females together and you get all 3 females giving birth, it's 100% fertility. If one gives birth, it's 33%. And there's 100% fertility. Cross a minus p53, minus male with a plus female, it's about the same. But now cross minus by minus, and it goes down to 0.69. Cross a plus male to minus, it's 2, right? Heterozygote to minus, it's down low, right? And the fertility rate goes down. So this says that you have to have the p53 gene in females, not males, in order to make, as you'll see, LIF, that's the hypothesis, right? But you have to have the p53 gene to have a wild type fertility rate and a wild type litter size. Fecundity, it could be regulated in this case in mice by p53. This is absolutely the same true for a different inbred strain, 129, with slightly different numbers, 5 to 2, for example, and minus crosses. And the cross of 129 by minus minus by black 6 gives also a small number 2 compared to the 6 and the 5. This is the production of Lif after mating of mice in the uterus of these mice. So here's day 0 at which copulation occurs. At day 4, which is the day of implantation, Lif is produced, and then by day 5 it's no longer produced. And that Lif had been shown to be absolutely essential for implantation of the embryo. Here's another p53 regulated gene in the uterus, and by day 4 it peaks and it stays up. Different kinetics, but these p53 regulated genes are all on in the uterus. Here's an example of what happens to Lif production in different genetic backgrounds. So this is day 0. D0 is in gray. In black is day 4. And this is the wild type black 6 mouse. And you can see there's about an 8-fold increase in Lif production. This is relative levels of Lif production in a wild type p53 mouse. Here's the heterozygote. That means plus minus p53. The basal level has gone down at day 0, and there's only a 2-fold production. And here's the null mouse. That's minus minus p53 female in a 129 background. Day 0, day 4, you can see it's at the same level as the basal level, and it doesn't increase to the high Lif levels. So p53 knockout mice are not making Lif in the uterus, and implantation is not occurring, and because implantation is not occurring, the litter size is low. And this is a good example of showing that. This is a uterus from a plus p53 mouse, and every one of these bulges is an implanted blastocyst in the uterus at 4 days or 7 days. And then here's what happens in minus minus mice. There are only 2. And here's the numbers for these experiments. For minus minus mice, about 2 on the average are implanted. For plus 8 on the average are implanted. But at day 3.5, just before implantation, there are 8 blastocysts, 8 to 9 blastocysts in both wild type and p53. So it's the implantation stage which is defective. Now here's the experiment that demonstrates, in fact, that Lif completely rescues this p53 genetic defect in implantation. So here is a minus minus mouse by minus minus, right, in black 6 and in 129. And then we either give a Lif injection or not, and we look at the litter size and we look at the fertility rate. Remember, the minus minus females are down at 0.69 per litter and a 27% fertility rate, but the minus minus females that are given an injection of Lif have restored to 6 in litter size and 100% fertility rate. And that's true for 129 as well. And this is just a wild type mice, right, given Lif or not, have about the same litter sizes. So that says that Lif can completely substitute for the p53 defect. And in so doing, this is the normal function of p53. Now, p53 was obviously described originally as a tumor suppressor that helped us respond to the stress signals I introduced you to. But this is a normal function of p53. It's not a stress-related function. p53 levels don't increase. And its normal function is, in fact, to act in the germline and to surveil implantation. Possibility is surveilling implantation, in fact, for genetic defects in the offspring. Right? Now, the reason I say that is that's exactly the functions of p53 in lower organisms. In flies and worms, p53 protein is only found in the germline, and it responds to DNA damage, for example, by destroying sperm or eggs, spermiogenesis or oogenesis, during the time period where defective organisms would have been formed. Right? So the question becomes, is there a surveillance mechanism here that is looking for defective offspring? And we actually have an experiment that can do that. We have a minus-minus embryo, we have a minus-minus mother, but we can give injection of Lif to rescue it. What do these embryos look like? And here's a couple of examples. They occur at variable frequency, but most of the time in C57 black, this is a newborn male. This is a newborn female. The females are runted and defective. They have defective neurological tubes. They have other defects. And this is an example in 129 background of a female and a male. There are males which are defective, but the sex ratio of defective offspring is decidedly in the presence of females, which brings up a couple of interesting possibilities that we're investigating: the possible role of imprinting, the sexually dimorphic effects of imprinting, or the possible role of genomic instability during development that p53 is surveilling. We know that if you have a pregnant mouse and you irradiate that mouse, that the offspring will abort by a p53-dependent mechanism, right, to get rid of the defective embryos. So the normal function, it looks like, of p53 had evolved in the germline to ensure the fidelity of development, ensure the fidelity of the genome, but was readapted, in fact, to fidelity in cancer cells. Cancer cells are notoriously unfaithful in reproducing their DNA. They make mistakes all the time. They have abnormal karyotypes, for example. And so p53 recognizes that and surveils it. Now, as we finished this work, when we finished this work, and as we were writing it up, we began to read journals we don't usually read. This is, I think, the American Journal of Fertility. And the journal that we were reading and trying to find out more about the work we were doing had a remarkably surprising article by Kulam and her colleagues from the University of Chicago. Dr. Kulam runs a in vitro fertilization clinic, and she's interested in implantation efficiencies because that's a rate-limiting step in vitro fertilization clinics. And for reasons which are less than clear, she looked at the single nucleotide polymorphism I've been telling you about in p53 at codon 72. Remember, proline has low activity, arginine has high activity, and here's the title of her paper. p53 tumor suppressor gene polymorphisms are associated with recurrent implantation failure. And the results, if you're a pro-pro woman, the female that's harboring the embryos, the frequency of is enhanced in 205 women with recurrent pregnancy loss in an in vitro fertilization clinic due to recurrent implantation failure. In other words, the mothers who are pro-pro, who make less p53, are presumably, based on what we've done in mice, making less LIF and having lower levels of implantation. So this is the beginning of finding a genetic correlation of what we found in mice and human beings. Let me finish by thanking a large number of collaborators who have made all of this work possible. First, my lab at the Cancer Institute of New Jersey, and I've mentioned Wenwei Hu and Angie Tereski, but many others, Kim Hirschfield, Elizabeth Bond, Sandy Harris, have been very, very helpful. The group here at the Institute for Advanced Studies, who largely did the statistical analysis and chipped in on the criticisms of the work we were doing, played a central role in the interactions between these groups. Louise Strong in the genetics clinic at MD Anderson and Gigi Lozano collaborated with us on Li-Fraumeni families. Ken Offord at Sloan Kettering collaborated with us in great detail on a number of different tumors. Frank Bartle and his colleagues in Germany and Halle collaborated with us on the sarcoma research. An experiment I haven't talked about with Ernie Beutler and Jill Whalen at Scripps, we actually looked at the relative risk of having the G allele over a lifetime, which was about 1.3, 1.4-fold relative risk over a lifetime for the G allele, but only in females, not in males, which fit all of the studies that we talked about. So let me say thank you very much. We'll stop, and I think I still have about a minute or two for questions, so we'll take a limited number of questions. Thank you. In fact, we'll take one question so we can stay on time if there is one. Yes?
Is this on?
Should work, hopefully.
Okay. The p53 and MDM2 pathway is fairly generic. What is your impression of studying cancer? Is it really type by type and tissue by tissue, or can you get a lot out of looking at these very general pathways that will go across all types?
I think like almost every bit of genetics, we're going to see a lot of differences. So I expect that since this SNP is regulated by an estrogen receptor, tissues must have estrogen receptors, although there's a surprising number that do. B cells, diffuse large B-cell lymphoma, have estrogen receptor. Lung has estrogen receptor. But of course, that limits this usually to females and usually to premenopausal females. The place where this SNP, I think, and we're just doing the study now, could have the biggest impact is that women who are postmenopausal that choose to take hormone therapy, estrogen-related therapy, go back into a higher risk group here by taking that therapy. And there is a slight increase in the frequency of breast cancer and other cancers in that group that takes estrogen, postmenopausal estrogen therapy. I don't think this one SNP is going to do it, but I think we could put together a panel someday that will mitigate, will be able to allow to tell a woman that she can or she should not be able to avail herself of estrogen or hormonal therapy postmenopausally with or without risk, or what the risk really is in both cases, right? So I think if I was to project in the future, I think looking at one SNP is probably not what will happen. It'll be a panel, and I think I try to stress that. Secondly, it will be useful in assessing risk, which is, of course, the topic of this particular session of the symposium. But there probably will always be risk. Genetics is probabilistic and interacting with the environment, so that one will have to try, as we heard in the first session, to explain to patients what the risk is. But these kind of studies give you quantitative data to do that.